Hello, everyone. Okay. I apologize in advance if I seem a little bit out of it. I just woke up from like one of those naps where you think you're in another dimension when you wake up. Like it was a powerful nap that I really needed. And then I was like, oh shoot, it's like 6:45. Let me go eat a cold taco and I'm here. So if I look like I just woke up, I did. So tell me guys how you feel about how you guys feel about the lab. So Tuesday, Thursday, you guys had your lab and it was the precursor. So you set up your lab number 7 and you guys also looked at some of your results for like the Msa and the Emb plates. So there's that. And then if you're Monday, Wednesday, you already saw your results for lab number 7. And you should have also seen your phenol tubes. So how do we feel on those? I'll write your guys' name down. And just wait a couple minutes to see if anybody else is coming. Okay. Did you guys almost, it's not bad, but did you guys almost forget that I was going to have a session at 7? Because I feel like I sent out the reminder maybe a little bit early. And I don't know, I feel like it was probably easy to forget between like 3 p.m. and 7 p.m. that, I don't know. Okay. Man, I felt like Wednesdays were always the packed day where everybody would come. Well, let's just jump into it, shall we? Let's see. So last week, let me take a peek at who was here last week. Okay, perfect. So we'll just finish up because you guys were there. We'll just finish up reviewing that quiz slide, and we'll finish reviewing that. And if you guys do have any questions, feel free to ask me. Sometimes also it just takes like recanting the information over and over again in order to like drill it, you know? So if you guys do want to just be like, can you just listen to me talk and tell me if I'm right or wrong? I can do that too. So last time we left off on coenzymes. So this is the next question. So this is coenzymes. So our next question is right here. do we want to take a stab at this one? So it says under ideal conditions, the complex aerobic oxidation of one molecule of glucose by a bacteria allows a net gain of how many ATP molecules? I've got a C. Oops, sorry. Okay, so what, I'm going to build off of this question. So what metabolic process is this specifically talking about? Gotcha. Catabolism. Okay, I think you're on the right track, but there's another word that I'm looking for. So this one is going to be in part of your lecture material. Let me go ahead and pull it up. I probably spelled that wrong, very wrong, but it's okay. I spelled it really wrong. Okay. Man, this is all really sciencey looking pictures. Let me just pull up the PowerPoint slide, and then I'll reshare my screen. So it's opening right now. What
Does it have to do with the Krebs cycle or like cellular respiration?
You're on the right track. So here, let me pull up the what specifically this thing is talking about. Here we are. So for this one, we have fermentation and respiration. So what the question was asking us was the complex aerobic oxidation of one molecule of glucose by a bacterium allows a net gain of how many ATP molecules? So if we look at right here, we're going to start at glucose and we're going to make our way down to our 2-pyruvic acid. Remember that glycolysis happens whether or not you have oxygen, and it can either go to fermentation or respiration if you have oxygen. So the complex oxygen-based metabolic activity that's happening is respiration. And so what this is looking for is what's the net gain of ATP from one glucose through the tract of respiration. So do you guys know what that is? I had a C, which I believe is to be correct. I'm gonna double check that. Yes, so C is correct. I just wanted to make sure that we knew why. And I only got one C, so. But yeah, so which one produces more energy, respiration or fermentation?
Respiration.
Yes. And glucose catabolism, right here, you're right with the bigger picture, but if this question is asking you which one is more correct, and it has catabolism versus respiration, respiration is going to be more correct, even though catabolism is still technically correct. I just wanted to show you guys that. And by the way, this definitely memorize. I can almost say without a shadow of a doubt, there's going to be several questions about this on the exam. Like anything that's kind of looks like a slide like this, like jot it down. Like another thing, this one. Like jot down the ones that you need to know, which is just I think it's that alcoholic, homolactic, and then the I think she said this one right here that causes the gangrene. It's what kind of happens with gangrene. I don't know if there's something like this. Something like this. Like a lot of the diagrams, if they help you, just redraw them. But yeah. So good thinking on this one. It's just usually that if a question asks about something like this, it's assuming that you know the background, and if you know the background, then it's going to ask another question if you like that. There's a million ways to ask a question about one topic, and it's almost impossible to figure out which one that they're going to use on the exam. So we'll move on to the next one. Most oxidation reactions in bacteria involve the, which one do we think this one is? Oh wait, am I sharing it right? I don't think I'm sharing it. Am I reshare it correctly? Okay. Which one do I think this is? And remember, on all exams, even if you don't know it, give it your best shot.
A.
I've got an A. Okay. Let me double check what the answer is. We got, yep, I believe you are right. A would be the correct answer. Do you want to explain how you got that?
I just remember oxidation has to do with losing electrons.
Okay, I'll take it. Yeah, it's remember the redox reactions. That was I hated that, the oil rig. I probably hated that the most in chemistry. So good job for remembering it. I wish I could forget it. So here is the next question. Sorry, I think somebody's calling me, but I got bigger, more important things to do. So I have the next question is anaerobic respiration typically uses as the final electron acceptors. So it's either organic or inorganic. What do you guys think?
Organic.
Organic, okay. I've got another chat. Organic, okay. So the answer is inorganic. Oh no. Okay. So let me find that answer. So it's aerobic respiration, right? Or anaerobic.
Right. I think I mixed the two up.
Yes, it's
aerobic uses oxygen, and oxygen is organic.
It's always got to be so similarly worded.
Yeah.
It's always got to be like a difference of a couple letters. And we love to see it all the time. Okay. So here, I'll share with you guys the specific place where I found this answer. Okay. So we have a nice little summary area right here where it's where it says the aerobic respiration, anaerobic respiration, and then fermentation in another spot. So what we were looking for was the final electron acceptor. And so oxygen, is that an organic compound, or is it an organic substance?
Oxygen is organic.
Okay. But then we also see over here in the anaerobic respiration that we have these inorganic compounds, and this one conditional one says externally acquired organic molecules. So these are the primary inorganic molecules you'll see, and then you'll see in fermentation that organic molecules is the final electron acceptor. So aerobic respiration is oxygen. Anaerobic is going to be, okay, is going to be an inorganic molecule, and then fermentation is going to be an organic molecule. So there we go. Let me get back here. And that one's kind of like you have to reach for that information. It's usually not one of the bigger ideas. It's sometimes it's kind of hidden, but that would be the reasoning behind this one. Again, I'm sorry if I'm so slow today. So we'll move on to the next one, a reduced molecule. Let's see, what is the best option here? Has gained electrons. B?
A.
A? Did I have a B and an A? Or were you changing your answer?
No, A.
A? Okay. I think this is another oil rig one. So is a gain. So yes. So I can see why people would say B, because you see it's gained something, and you might think it would be a charge. But yeah, just remember, I'm gonna double check it before but just remember, electrons negatively charged, but you caught yourself. That's what counts. All right. So that is the end of this review one. I know we were so close to finishing it last time. I think we only had like 4 that we needed to do. So I'll put that away. Are there any, so do we understand what MSA, EMB, what those plates do, how they function, what makes them selective versus differential, maybe how to read them? Do we understand what we set up or saw in our lab number 7, our fermentation lab? And so many power searches. Or are you guys good on that? Don't really need review.
I didn't. I wasn't there for lab 7, so can you go over that?
Yeah, of course. So lab number 7 is the fermentation lab, and what we did was we took grape juice, right? We took our grape juice and then we put it in a flask and we added yeast to it. And then we put a balloon over the top. And then we also took a little Dixie cup, put some boiled milk inside of it with a dollop of yogurt, and covered that up with some foil, and then also left that to kind of settle. So that was the gist of lab number 7. So let me pull up the because it'll help me kind of visualize it for you guys. It always gets like really small. Like I don't know why that happens. It's like this is the slide. It's not like a preview slide. This is the slide. I don't know why it does that. It's kind of funny. Okay. Let me get back to the fermentation area. Here we go. So we with our grape juice, we found that we were going to do the alcoholic fermentation. The point was to make wine. So what we had to do was we had to take a sugar source, right? In the grape juice was glucose. It's like half sugar. Like there's a lot of sugar in like Welch's. So we took our glucose in our grape juice and we took an organism that would be able to do this fermentation pathway. I remember what is yeast? Do we remember why it's so special? You want to take a jab at it? So I wanted to double check that I was giving you guys the right answer. But yeast is special because it's technically a mold, but because molds are multicellular, this one's special because it's considered a unicellular mold. So that's the special terminology, nomenclature that you would use in order to address yeast. It's a very popular, posh little organism we have at our disposal. So we took our glucose and an organism that's able to perform glycolysis and fermentation. And so we put them together and we were able to go down this alcoholic fermentation pathway and produce ethyl alcohol. That's what is muy delicioso. That's how we make the wine and several other alcohols. But we also put the balloon over the top. Why? Because another byproduct of the alcoholic fermentation process is CO2. And you're going to see that balloon on top inflate. And then once you pull it off, it's going to smell like wet red wine. You can roll it in your hand, but don't drink it. Like it's not going to be good for you, but you can smell it if you really wanted to. It's probably going to be really bitter, or like disgustingly sugary. There's probably no in between, but that's what we saw when we wanted to make our wine. So then we move on to our boiled milk and yogurt. So our boiled milk has what sugar? Lactose. Yeah, you get the runs if you don't like that stuff, if your intestines don't like it. And I know it tastes good, but get those lactase pills. Like don't torture yourself if you're lactose intolerant. But we have our sugar source, which is lactose, and we introduced the probiotics in the yogurt that were able to perform glycolysis and the fermentation pathway. So what happened was we put those two together, and the reason why it's boiled is because it gives the bacteria a nice little cozy feeling. Like, you know, if you take a shower and the water's cold. You're not gonna want to stay in there, and you're not gonna want to do anything. Like you're gonna wait. Like it's gonna take forever for the water to get warm and for you to finally take a shower, and then you're gonna be like, oh my gosh, why am I even here? That's kind of the same thing with the probiotics and the yogurt. They want to make sure that they're walking into a nice steamy shower. They want to get somewhere warm and cozy before they even start doing anything. So speaking of which, I'm gonna go take a shower after this, but it sounds so nice. I digress. So we have our lactose sugar. The probiotics in the yogurt are able to perform glycolysis and do this homolactic fermentation pathway. And that produces lactic acid. So what are we gonna see in our little Dixie cup? We're gonna see curdling. We're gonna see a little bit of cheese. It might look like kind of a cottage cheese little thing going on. Do you guys like cottage cheese? I feel like an old woman if I say I do, but it's good, no shame. I like cottage cheese, especially if you put little fruit in there. It's really good. Anyways, but that's just the gist of lab 7. Let me double check that I'm not missing any key ideas. Got my Bible. I don't want to bring this in person because it's a little intimidating, but I've got plenty of information in here. Let's see. Where are you, lab 7? Also, I wanted to remind you guys that on Friday, I believe, there is going to be a practice lab available for you guys from 1 to 4. I think Dr. B and I might be a little caught up for the first half hour. We actually have a little bit of we have like a faculty meeting, but we will be there to help you guys walk around. Use the pre-made slides. You can gram stain. You can do whatever you'd like, but it's an opportunity for you guys to practice your lab skills that you'll have to perform in exam. So if you guys don't feel too confident for your streak for isolation, or you want peace of mind, it's there for you. It's available. And you're more than welcome. Even if you want to just not even touch any of the lab stuff and just ask me or Dr. B questions, you can do that too. Let's see. Alcohol fermentation, milk fermentation. Don't forget, you guys will need to do a chart for this one, I believe. There's also a nice little simplified fermentation pathway chart on page 72 if yours is still the same as mine was. Let's see. And then you'll have to answer some questions about the different parts of either glycolysis, fermentation, or respiration. You'll have to answer some questions about that. And I believe that is it. That is most of the plate. Most of the lab. Do you guys understand the phenol tubes, by the way? I'll scroll down to these. I think, yeah, these are the phenol tubes. So obviously they don't really look like this in our lab. It's a little less apparent that there might be gas, but do we understand the 4 components of an F tube?
Can you go over that too?
Yeah, of course. So our F tube has obviously 4 components, like I said. We see a difference between this pink one and this yellow one because there's the phenol indicator. It's just like your MSA plate. There's a phenol indicator there and just a phenol broth, same thing. So it's the pH indicator that we're going to see. That's going to change it from pink to either a deeper pink if it's producing more basic byproducts, or no change is going to look the same as the original tube. And then we'll also have our bright yellow, very distinct acidic byproduct. So that's the phenol portion, and that's how it functions. And then we also have our Durham tube, right? It's right here at the bottom. Our Durham tube allows us to see gas production. So that's just going to tell us a little bit more about what exactly this metabolic process is, like what exactly is happening, right? What metabolic process is this bacterium going to go under? So the Durham tube allows us to see gas production, and that's technically one of the ingredients. And then we also have a sugar source, right? Remember how we had so many tubes? It's because each one had a different sugar source, or each of the three, I think. Was it 3 or 4? No, it was each of the groups of 4 had different ones. So we had lactose, we had sucrose, and we had glucose, right? Lactose, sucrose, glucose. Yes, and another word for glucose is the dextrose, right? So that's going to be another component. It's not all 3, it's one or the other. It's one of the 3. So we have the phenol red, Durham tube, and the sugar, right? And then we have a last one, and that's going to be amino acids, right? We're going to see the more basic, deeper red if the amino acids are used and not the sugar. And that shows us that it's not fermenting. If it's not using a sugar as a sugar, or it's not using the sugar as the food source, then it's not fermenting. It's using the amino acids as its energy source. So we just give the bacteria an alternate to see which one they prefer. So those are the 4 components. You got phenol, Durham tube, sugar source. It's either sucrose, lactose, or dextrose / glucose. And then we have our last one. It's going to be amino acids. So phenol red, Durham tube, sugar source, amino acids. Okay, does that make good sense? And then I believe that, yes, so if we see that this, like the tube on the left, it's going to have a, if it's going to appear pink, we're going to put a K for a negative reaction, because the whole point of the F tubes is for fermentation, F fermentation. So if we don't see fermentation, we're going to put a K. So K just means that it's basic, no reaction, red is negative, right? But if we see a yellow positive reaction, we're going to put A for acidic. Don't ask me why they decided to do K instead of B, like for basic. Like, I don't know. Like, just ask some professor 100 years ago who's probably bald. They have bigger problems because they're bald. But we have our acidic tubes, and then our basic tubes. So A acidic, basic is going to be K. And then if we see a gas production, we're going to mark G, which, 2 out of 3 makes logical sense. But the third one, why? I don't know. So for the left one, what would we indicate? For the left tube here, the completely pink one, what would we indicate for that one? It's just going to be letters. It's nothing too fancy. K, good job. I see where you were going with the N, but I see it's K. It's K, right? What about the middle one? What do we see there? We see acid, and we'll just mark that with an A. There you go. It'll be a capital A, but you guys get the gist, and I understand. So the middle one, we would just indicate that that has an A. And then for the last one, what would we do? Remember, A stands for acid, and then, yep, Ag. Perfect. So that's just how we mark these. And they're relatively straightforward. If it doesn't have a gas, you're not gonna put like no gas. You're just not gonna put a G. You're just gonna put K or A. But yeah. I think so. That's all basically of lab 7. It ties into our MSA and EMB plates because remember, those are going to be possible. It indicates possible fermentation, right? If we see that there are purple, pink, or green colonies on an EMB plate, we see that they're lactose fermenter, right? And if we see the phenol indicator change on the MSA plate to yellow, then we know that the substance is going to be a mannitol fermenter. Okay, so connecting the dots. It's like a constellation in the sky. Okay, so how do we feel? I'm sorry if it's a little all over the place. My brain's playing ping pong with the last 2 brain cells I have right now. But yeah. So I am going to, if you guys don't have any questions right now, and I'm not leaving you hanging, I'm gonna go ahead and let you guys have like a 5-minute break, get some water, think of any questions whatsoever. I don't mind talking about even study habits, the format of the test, I've taken them. I don't mind talking about, I don't know, anything really. It doesn't have to be lecture or lab material. And if you guys are in Bio 33, by the way, I know you guys have your exam coming up because they basically land almost on the same days. So if you guys need any help with maybe some of those concepts, I can kind of assist you through those. But yeah, so I'm gonna let you guys go take a 5-minute break. It's 7:38, but I'll just make it kind of 6. So at 7:44, go ahead and come back and rejoin the meeting because I have to stop it and then I have to start like a new one because it's like you can only do 40 minutes. So I'm gonna follow the Zoom gods and obey them. So go ahead and rest, relax, think of any questions you'd like, and I'll be here and open up a new meeting at 7:44, okay? If you guys also need to go, that's completely fine with me. I'm gonna end it. I'm sorry, I'm kicking you guys all out.